Hey Team Bio, I heard my last screencast was a little muffly at some points. I will try to be a little bit better about not putting my hand over the microphone. Actually, I don't even know where the microphone is. Oh, it's over there. Okay, I think. So I won't put my hand on that side of the iPad. Okay, so yesterday you had a screencast. It was an introduction to cell respiration. Just a reminder, this is all content that will be on a quarter 4 test, not on the upcoming test on Friday. But this is just to get us prepped for when we come back from break to dive into cellular respiration. And we've talked extensively about the chemical equation for photosynthesis, and then how that relates to the chemical equation for cell respiration, and we get a flow chart overall of cellular respiration. And today's screencast is going to be focused on glycolysis, which, remember, occurs in the cytoplasm of the cell. We'll be getting to what happens in the mitochondria later when we talk about aerobic cellular respiration that happens in the presence of oxygen. But I want to introduce you to a concept that's pretty important in both photosynthesis and cell respiration, but we didn't really talk about it directly in photosynthesis. And this is an idea, it's called a redox reaction. And this is a concept you might have learned about in methods, and you'll definitely talk about in chemistry. But a redox reaction is a shorthand for a reaction that is a reduction oxidation reaction. So it's a reaction that comes in a pair where one thing is reduced while the other thing is oxidized. And reduction and oxidation is basically the exchange of electrons. And an acronym that helps me remember which is LEO goes GER, where LEO stands for loss of electrons, oxidation, gain of electrons, I guess I should put the of here. Loss of electrons oxidation, gains of electrons reduction. So whenever something gains electrons, it must have taken them from somewhere. So the thing that is reduced gains the electrons. A little bit confusing there. I would think that if you're reduced, you're losing something, but you're actually gaining electrons. I guess negative charge. So you're smaller. You're not. Anyway, I don't know why it's called reduction oxidation. And when you're oxidized, you have a loss of electrons. So this reaction always comes in pairs, redox reaction, because if you gain something, it's got to come from somewhere. If you lose something, it's got to go somewhere. So a redox reaction is when we're going to move electrons from place to place. And this is important because, remember, photosynthesis was all about, when we boil down to it at its root, we were stripping low energy electrons from water. We were exciting them using energy of the sun, and we're using them, we were fixing them, we were fixing carbon to make glucose, but we put those high energy electrons into this molecule. So glucose is storing our high energy electrons here. And cellular respiration is all about extracting that energy from glucose and moving it to now ATP. So we're going to pull out high energy electrons from glucose, and ultimately we're going to use their energy in order to do something, make ATP, and ultimately those high energy electrons are going to become low energy. We're going to spend their energy, and they're going to end back up on water. So it's kind of this beautiful symmetry where we strip the low energy electrons, we make them high energy, we store them on glucose, we pull them away from glucose, we use them to do something, and then we throw them back on water at the end. So Redox reactions are going to be super important. They're important in photosynthesis, but we're going to be paying close attention to them in cellular respiration. We're going to be looking what happens when our electrons pulled off of glucose, and ultimately, where are they going? Okay, so starting with glycolysis. So glycolysis, glucose, lysis, meaning splitting. The whole point of this reaction is to split glucose up. And in the process, we're going to strip some electrons away. And we're also going to make a little bit of ATP. So we start out with our 6-carbon compound, glucose. Now, as we talked about in the light-independent reactions, these carbon molecules that we're talking about are much more complex than what I'm drawing here. You know, this is actually C6H12O6. Oops, that was not a 6. O6. But we're only going to focus on the carbons here. So when you take biochemistry in college, you'll have to learn the molecular formula and structure of all of these intermediates, but in our class, we just care about what's happening to carbon. Okay, so we start out with glucose. That is what goes in the glycolysis. And remember, this is all happening in the cytoplasm of the cell. Glucose needs to be actually charged up before we can break it down. So we're going to charge it up by adding 2 ATP. The phosphate groups of these ATP are actually going to transfer onto glucose. So I think I want some dotted lines here. So our ATP is going to swing in here, and the phosphate groups from this 2 ATP are going to be transferred to glucose. So in doing so, we spend, whoops, I think I want this actually to be not a dotted line anymore. We spend 2 ATP, and ultimately, you know that when ATP is broken down, we end up with ADP, so we end up with adenosine diphosphate because we've transferred 2 phosphate groups here. And because we're spending ATP at the beginning of this reaction, we call this phase of glycolysis the investment phase. We actually have to use a little energy to get this reaction started. Next up, we have a redox reaction. Oops. So let's pay attention to where the electrons go. This 6-carbon molecule breaks down into 2 3-carbon molecules when 2 more phosphate groups are added. I think I want it to be this dotted line again. So there are just 2 free-floating phosphate groups in the cytoplasm, and we're going to add 1 here because we've broken up this carbon molecule. So adding this phosphate charges it up. We can break it in half. Now we add another phosphate here, and we add another. We add another phosphate here, and now we've got 2 very high energy 3-carbon molecules. This is called, I want to go back to these 3-carbon molecules are called G3P. And now G3P is going to be oxidized in this process. This means it's going to lose electrons. So the electrons are stripped away from G3P, and maybe we should show them as dotted lines. I don't know. So we're going to strip out electrons from G3P. And remember, these are high energy electrons that were brought to glucose, back when it was glucose here, in the light independent reactions, and they were carried by a special carrier molecule to the light independent reactions called NADPH. a very analogous molecule comes swooping in here and picks up these high energy electrons along with some H plus to form NADH. So this is reduced. It's grabbed high energy electrons and it's carrying them. And so gain of electrons is reduction, NADH is reduced in this process, while G3P is oxidized. Now, this molecule is going to hold on to these high energy electrons because they're dangerous. If they're loose in the cell, they could just spontaneously cause a reaction to occur. So we need to keep them safe and hold them in this carrier molecule. We're going to use them much later, not in glycolysis itself at all. But we're going to need the presence of oxygen in order to use them. However, okay, so NADH is holding on to our high energy electrons for us. We've oxidized G3P. Now we're ready to proceed on to the next phase of the reaction, where we're going to perform the payoff phase. Okay, at the beginning of glycolysis, we invested 2 ATP in getting it all started. Ultimately, we are going to swoop in here with 4 ADP and perform what's called substrate level phosphorylation. So you all have been comfortable, or not comfortable is the wrong word, but you've been exposed to the idea that an ATP synthase machine, protein embedded within a membrane that has a concentration of hydrogen on either side can perform phosphorylation of ADP into ATP. But there's a second way that ATP can be made, and that's just when we bring ADP over to kind of touch a molecule that contains phosphate groups, and we strip those phosphate groups away. So everything that we just put on to glucose, or what is no longer glucose but is now G3P, all of these phosphate groups are going to be pulled off of the molecule. And oops, I guess I want to do dotted lines again. Now, they're all going to be transferred onto ADP to make it ATP. Okay, that's kind of a little messy, but you get the idea. So we're pulling off the phosphate groups that are directly attached to this molecule, and we're adding them to ADP to make 4-ATP. So that's called substrate level phosphorylation, because this is getting phosphorylated. We're adding a phosphate group to it, and it's substrate level because it's happening at the level of, this is our substrate. There's going to be an enzyme that binds to G3P and does this. Okay, so the net payoff from glycolysis is back to red here, back to bold, 2 ATP are made, and 2 NADH are made. Okay, and that's it. Out of this process is a 3-carbon compound called pyruvate. And pyruvate has a forked path here. In the presence of oxygen, it's going to go down the aerobic pathway, which is going to be much more lucrative for the cell in terms of energy extracted from the molecule. And then in the absence of oxygen, so if there's no oxygen, we're going to form anaerobic cellular respiration, and we're going to proceed to fermentation. So your next screencast is going to be an introduction to fermentation, and you're going to see how that all works. Okay, that's all.